Alright, so for our second installment on microbial metabolism, alright, so we'll focus on just enzymes. Alright, so again, this material should be review material, so this is a refresher. Alright, now some of it towards the end will be new material. Alright, but majority of this is just going to review the main concepts of enzymes, what they are, how they function, and what they do. Alright, so first off, giving you a little video on how enzymes work. Alright, so when you're talking about enzymes, now, just the protein portion of an enzyme, alright, is the apple enzyme. Alright, so conjugated enzymes contain proteins and a non-protein molecule. Alright, so this is the whole enzyme. The whole enzyme is going to be the functional enzyme. Alright, so this whole enzyme you can break apart into apoenzyme and cofactors and coenzymes. Alright, so the non-protein molecules it's talking about are your cofactors and coenzymes. Alright, so the apoenzyme is just a protein. Alright, the cofactors are organic molecules, alright, and coenzymes are inorganic elements, metal ions. So the organic molecules would be things like your vitamins. Alright, so basically your vitamins are basically, majority of the time, your vital amino acids. Now, an enzyme has a specific site for which a substrate molecule binds. Alright, so this is referred to as the binding site of that particular substrate, or the active site. Alright, it's referred to the active site as the active site because this is where the activity occurs. Alright, so this is where you have the interaction between your substrate and your enzyme. And this is also where you have the interaction if you have, like, for instance, you have your protein. Let's say you have a binding site for reactant A. You have a binding site for reactant B. All right. Now this is where you have the activity for A and B that are binding to the enzyme. This is also where you have the activity where A and B come together to form your product, AB. All right. For instance. Okay. Alright, so this is a site of biological activity on a protein. Alright, it's the active site. Alright, so here you have your enzyme, you have your substrate molecules. Now notice, your substrate has many different parts to it. Alright, different little puzzle pieces, jigsaw pieces, that necessarily won't fit in this particular binding site, alright, but one might actually fit. Like this one fits here, and this part fits here. But then notice, this part on this substrate binds to this part here on this other substrate. They come together. Alright, it's the enzyme. What is it that an enzyme's doing? Well, the enzyme is basically this docking station where reactant A and reactant B can attach to the enzyme. And the enzyme can bring these two guys together, alright, to form your overall product. Alright, so basically, if you're talking about a chemical reaction taking place without an enzyme, alright, you have to wait for reactant A and B to come together and collide in such a way that, right, this part of this substrate and this part of this substrate actually come together. They come together with the right amount of energy, they come together with the right amount of spatial orientation so that they fit, and so they have the formation of that chemical bond that holds them together. Alright, so the enzyme facilitates the process. Alright, so the enzyme does not participate in the chemical reaction, but it facilitates it. Right? It helps it take place. Alright, notice that when your substrates bind, notice how the enzyme changes its shape. Alright, the enzyme will actually kind of wrap itself around and conform around its substrate molecules. And what it's doing is it's bringing the chemical groups of these respective molecules, alright, A and B, in close enough proximity and orientation for where they can interact. Alright, so you can form your new product. Alright, so again, looking at some terminology. Alright, so you have your apoenzyme, which is just the protein part here. Alright, you can have cofactors. Alright, you can have coenzymes. You have your binding site for your for your substrate. Alright, now this entire thing, right? So basically, your holoenzyme is basically your protein plus your cofactors or your coenzymes. Alright, so if you're talking about an enzyme without its cofactors or coenzymes or cofactors, it's not going to be functional. Alright, it requires these things to function appropriately. Alright, so again, coming back to this idea of cofactors and coenzymes. Alright, so enzymes don't act alone. Alright, so here you have your apoenzyme. Alright, and here you have your holoenzyme. Alright, because you have your cofactor plus your protein. That makes your holoenzyme. Alright, so cofactors are things like ions, metal ions, so zinc, magnesium, iron. Your coenzymes are organic molecules, typically vitamins, but they can also be involved like NADH, FADH2. Alright, so your cofactor binds. Notice how this cofactor has a shape that's going to conform to the shape of the protein. It's like a saddle. It sits on there in this example. And then once it sits on the protein, the binding sites, the active sites for the substrates, open up. So now this protein is active. The enzyme is active. Alright, so when you're looking at enzymes, alright, there's a lot you can learn from the name of an enzyme. Now we'll pick back on the naming of enzymes towards the end of this lecture. Alright, but notice here, alright, you have sucrose, alright, which is a disaccharide, that's being broken into glucose and fructose. Alright, both of these are monosaccharides. Alright, so in this case, the enzyme isn't involved in a synthesis reaction, it's actually involved in a degradative reaction. Alright, so in this case, a catabolic reaction. Alright, so this is a hydrolysis reaction. Alright, so your sucrose molecule will bind to the enzyme sucrase, and the water molecule will then break off that covalent bond, alright, the glycosidic linkage here, it'll break it, and you attach a OH group to the glucose and a hydrogen atom to the fructose molecule. Alright, so hydrolysis. And you end up with your products. Alright, so what exactly is taking place here? Well, your sucrose molecule binds to the enzyme sucrase. Sucrase will wrap around that molecule of sucrose. It will bend and orient the molecule so that the oxygen atom here is exposed so that the water can come in and break it. So you end up with your products. Alright, so the enzyme facilitates in basically kind of torquing and twisting that molecule of sucrose. It weakens that covalent bond so that it's easier for that water molecule to break that covalent bond. Also notice the name of the enzyme, the prefix, alright, sucrase, sucra, alright, resembles the name of the substrate. Alright, this isn't always going to be the case, alright, but in this instance it is. But one way you can distinguish a substrate from its enzyme is you look for the suffix ASE. That's how you know that this is an enzyme. Alright, and this is your substrate. Alright, so here you're being shown kind of an animation of an enzymatic process taking place. Alright, so here in the animation you have your products release, or you have substrate 1, substrate 2. Right? They bind to their active site on the enzyme. Right? You have an enzyme substrate complex, you have the reaction, you have the covalent bond that forms between your reactants to form your product, and your product's released. Alright, so your enzyme facilitates this reaction. It helps it take place. So that means that your enzymes, alright, can be recycled. They can aid in many chemical reactions. Alright, so here, alright, you have two substrate molecules that bind to the active site of an enzyme. Now notice, when these substrate molecules bind, the shape of the enzyme is changed. Alright, because you have this enzyme substrate complex. Alright, so the enzyme actually wraps around and kind of holds those substrate molecules in place. Right? And it's also bringing the chemical groups of your substrates that are actually going to be involved in the chemical reaction of bonding to one another together in close proximity and the right orientation. Alright, so next, what's happening is that every single chemical reaction, alright, there is something called an energy barrier, alright, a threshold. that a chemical reaction has to get past to actually take place. Alright, so the enzyme itself is actually lowering this level of what we call the E sub A, or the activation energy. Alright, so this is how it's speeding up the reaction. Alright, so imagine you didn't have an enzyme. Alright, that means that these two substrates in the flask have to collide with one another in the right orientation, with the right amount of speed, with the right amount of energy, so that they actually come together and form a product. Right? Now that would take a while. It would take a while to end up with the same amount of product you would get using an enzyme. Alright, using an enzyme is going to speed up this process. Alright, so here, your substrate molecules, right, you have the chemical reaction that takes place, and your products are released. And now the enzyme can go back in and bind to new substrate molecules. Alright, so the enzyme is neither a reactant nor a product. It just facilitates the chemical reaction. Alright, so here, you're being shown a chemical reaction. Alright, so it's a chemical reaction. You have A, reactant A, and reactant BC. Alright, so here, what's going to take place is B is actually going to be transferred from BC to A. Alright, so you have a substitution reaction. Alright, so you're transferring one molecule from one component of a molecule to another. Alright, so here, the level of energy is required to actually break this chemical bond, that covalent bond, and allow for the formation of this new one is very, very high. Alright, so your level of activation energy is very high. Alright, so the cell would have to input a large amount of energy, alright, so that you're breaking this bond and you're forming a new one. Alright, so the reaction would reach what we call this transition state, alright, where you're in the middle of breaking bonds and forming new ones. So this would take a tremendous amount of energy to reach this level of activation energy. Alright, but once it's reached, alright, the reaction rolls downhill. Alright, and you'll end up with your product, AB plus C. Now, look at the reaction with an enzyme. Alright, so here, with an enzyme, the level of activation energy is much lower than without it. Alright, because what you're doing is you're bringing these two reactant molecules together when they bind to the enzyme at their active sites, and the enzyme will bring these molecules in a close enough proximity to facilitate and aid in this reaction. Alright, so that it can reach this transition state where you're breaking bonds and forming new ones, and the reaction will go downhill. Alright, so enzymes lower the activation energy needed for a chemical reaction to take place. Now, one thing is important here. The amount of free energy change. Alright, so notice that this is actually a exergonic reaction. Alright, so even though an exergonic reaction releases energy, it still needs a little help. Alright, it still has an activation barrier it has to get over to move forward. Alright, so the cell has to input energy in, a little bit of energy, to kind of get the reaction moving. Alright? But notice that the amount of energy that's actually released in this reaction, whether you have an enzyme or you don't have an enzyme, the amount of energy that's released stays the same. Alright, in this case, the delta G value is actually negative. Alright, so you get a negative delta G. So it's an exergonic reaction. It's a spontaneous reaction. Alright, but even spontaneous reactions need help to actually occur. Alright, so this kind of models what that figure, those past couple of figures are talking about. Alright, so you have reactant A and reactant B, C. They bind to the respective enzyme at their active sites. Alright. Now, your enzyme will actually alter its shape so that what it's doing is it's actually straining this covalent bond to weaken it, and it's also orienting this reactant molecule B so that it's in the right spatial orientation and configuration to interact with A. So now the cell, the reactants, have oriented themselves in the proper spatial arrangement so that the reaction can kind of move forward. Alright, so now you have the formation of this new chemical bond here between A and B. The bond that links B and C is broken. Your products are released, and your enzyme goes back to its normal shape. Alright, so enzymes, alright, they're referred to as biological catalysts. Alright, that's another word for them. Alright, so if you look at a chemical reaction, alright, you look at an uncatalyzed reaction and you look at a catalyzed reaction. Alright, notice that the speed of the reaction is much faster in terms of product formation with that of a catalyzed reaction, but an uncatalyzed reaction, it'll take place but at a much slower rate. Also notice that it's also a linear progression. Alright, your catalyzed reaction kind of has this logarithmic growth curve shape to it. Alright? But eventually, notice that it does begin to kind of wane. It levels off. It kind of reaches what we call a threshold. Alright, so basically, what this means is that you're going to reach this threshold because in a chemical reaction, you've added a finite amount of your reactants, and you've also added a finite amount of your enzyme. Alright, so eventually the enzyme can only speed up the reaction so fast. Alright, because there's going to be a point in time where every single enzyme that you have in your solution, alright, wherever the reaction is taking place, that every single enzyme has reactant molecules bound to it. Alright, so it can no longer bind to anything else. Alright, so your rate of your reaction, of product formation, will actually level off. Alright, the only way that you can kind of get this to keep going is if you add more enzyme. Then your rate of product formation will go up. Alright, so your enzyme becomes kind of a limiting factor in that case. But then again, so does some of your reactants, because once you use your reactants to form a product, alright, they're no longer available. Alright, so enzymes are proteins. Alright, most enzymes are proteins. Alright, there are RNA molecules that act as enzymes. Okay, so not all not all enzymes are actually proteins. Alright, so in terms of protein denaturation, alright, so there are various chemical, physical, mechanical factors that can lead to the denaturing of a protein. Alright, in this case, a denatured protein goes from its tertiary, either its quaternary or its tertiary state, to its basically primary state. It's unfolded. It's unraveled. And it's no longer active. And actually, when it has unraveled, you can have interactions within the protein where it starts kind of binding in on itself, and you get this inactive form of the protein. Now, if conditions go back to normal, your protein could refold, alright, and go through this process of renaturation to where it will be functional again. Alright, so when you're looking at enzymes, alright, here you have the activity of an enzyme. Alright, so the activity of the enzyme is measured as, in this case, the rate of the chemical reaction. Alright, so looking how enzymes function optimally, or at their best, alright, so the most optimal conditions for an enzyme. Alright, each enzyme is specific, alright, in the sense that enzymes in one type of organism may not function as well, or even at all, in another organism. Alright, so here you have two enzymes, and you're looking at the effect of temperature on reaction rate. So here, notice that for humans, alright, the optimum temperature is around 37 degrees Celsius. Well, this makes sense because that is human body temperature. Alright, so our enzymes work most efficiently at our body temperature. Now, If you look at a bacterium, all right, that lives in, for instance, like hydrothermal vents, or lives in warm, hot ponds. All right, so here the optimum is around 77 degrees Celsius. All right, so there's a shift in the efficiency of an organism that lives in a warmer environment. Their enzymes work much more efficiently than those at that warmer temperature, in comparison to our enzymes work most efficiently at 37. All right, so that's mainly because, think about it, bacteria, they can't regulate their temperature. They're totally solely dependent on the environmental conditions to determine the temperature. All right, so it's just they dwell in environments that have that temperature range, and their enzymes function best at that temperature. All right, so looking at the human enzyme. All right, now notice that as you go past 37 degrees Celsius, there's a bit of a limit here. All right, here and here. All right, somewhere around the 40 to 43 mark. All right, the rate of enzyme activity drops tremendously. And then it's basically no longer active at like 50. All right, so what's going on here? All right, well, think about it. You're heating up a protein. All right, so as you're heating up this protein, what are you doing? You're breaking the bonds that are holding it together. All right, so you're breaking the hydrogen bonds, you're breaking other forms of bonds, and the protein is denaturing and unraveling, and it's no longer active. But look here, for your bacterium at that same temperature, their enzyme's functional. And as you increase the temperature, the rate of chemical reactions increases. It's most efficient at 77 degrees Celsius, and then it drops somewhere around 90, and it's no longer functional. Again, their proteins can denature. Now, what is it about the lower temperatures? All right, why do you see this drop as you decrease the temperatures? Well, this has to do with the movement of molecules. All right, so as you're cooling the temperature down, what are you doing? The molecular motions slow down. All right, molecules still vibrate. Even when they're frozen, they vibrate. All right, but here you don't have enough thermal energy required. All right, so again, the rate of reactions goes down to 0. Now, this is looking at temperature. What about pH? Well, you have for instance, two enzymes in the human body. You have pepsin and you have trypsin. All right, so pepsin works most efficiently at a pH of around 2, no less, and trypsin around a pH of 8. Now, notice here that as you increase the pH, as you become more alkaline, all right, the rate of enzyme activity drops to 0 around 3.5, 3.7. All right, even though this is still acidic, all right, you're talking about the stomach, and so usually the stomach has a pH between 2 and 1, 3. It varies throughout the day. All right, mainly it's 2. All right, so again, as you increase the pH, you're also going to be denaturing the enzyme, because by altering the pH, what you're doing is if you have any type of ionic bonds that hold together a protein, those particular ions, their charge can change. So they can go from, if they're negative, they can go to neutral or positive. Those that are positive can go to neutral or negative. And so then that chemical interaction that was holding that protein together is no longer there as you alter the pH. Alright, so trypsin functions best in the intestine, small intestine. Alright, so a pH around 8. So notice here that, think about it, as you ingest food, the food makes its way through your stomach. Alright, the stomach is exposed to pepsin, and then as it makes its way into the small intestine, you do have a change in pH. Alright, the opening of the small intestine is still slightly acidic, but as you work your way through the small intestine, it becomes more and more alkaline. Alright, so this is where trypsin is going to be most active. And then, again, as you increase the pH closer to 10, enzyme activity just drops. Alright, so again, the enzyme is denatured. Alright, so enzymes have optimum temperatures at which they function best. They have optimum pHs. They have optimum salinities, optimum pressures. And these optimal conditions are tied in with their host and, a lot of times their environment. Especially if you're talking about a cell or a single-celled organism like a bacterium. Alright, so talking about enzyme regulation. Alright, so not every single gene in the DNA is expressed all the time. Alright, it's a very inefficient process if you were to do it that way. Alright, so when it comes to bacteria, and we'll hit on this later when we come back to this idea of regulation of the genome. Alright, but bacteria are very, very efficient in what they turn on and what they turn off, what they need and when they don't need it. Alright, now in terms of enzymes, you have two different groups. You have those that are what we call constitutive and those that are regulated. Alright, so constitutive enzymes are those that are always turned on. They're always expressed. Alright, so regardless of the level of a substrate that is in the environment, it's always going to express this particular enzyme. Alright, and a lot of these constitutive enzymes are basically about core functions for an organism. Alright, so think about it. Things that the cell always is going to need. Alright, so it's always going to need glycolytic enzymes. Alright, so central carbon metabolism is always going to need those enzymes. Alright, because when you talk about central carbon metabolism, it's kind of the conduit for other biochemical pathways. They feed into this, and other stuff can shoot off out of it. And it's just all interconnected, where things from central carbon metabolism can go to other areas, or other things can be diverted to central carbon metabolism. Now, that'll make sense later on when I talk about cellular respiration. Alright? But it's a huge interconnected network of metabolic processes. Alright, so ribosomal RNA, well, you need ribosomal RNA and ribosomal subunits to make your ribosome. Alright, so you have to be able to synthesize proteins. Alright, that's a needed function, a needed cellular function. Now, regulated enzymes. These are what are turned on. Now, if you turn on the production of an enzyme, it is induced, and if you turn off the expression of an enzyme, it is repressed. And this is in response to the concentration of a particular substrate. Alright, so here you're given two figures. Alright, so one figure is showing you constitutive enzymes. You have a certain number of enzymes that are always produced. They're at the same level, pretty much all throughout the the life of the cell. That doesn't mean that those level of enzymes can't be ramped up, but there's a basal level at which those enzymes are always produced. Okay, so it doesn't matter how much substrate you add, you're going to have the same number of enzymes. All right. Now, regulated enzymes, as you increase the amount of a particular substrate, for this instance, the level of proteins that are the enzyme in this situation are increased. So they can interact more with the substrate. So this would be an example of what we call induction. All right, so it's turned on. Now, here, if you remove the substrate, all right, think about it. If you remove the substrate for the environment, there's no reason for that bacterium to produce that particular enzyme if it's a regulated enzyme. If it's not going to be interacting with anything in the environment, it's a wasted process to make that protein. It's very inefficient. Alright, so this is an example of repression, where you decrease the amount of concentration of that particular enzyme, or the particular substrate, the enzyme concentration can be reduced to a very small number. Alright, it's repression. Alright, so there are two main ways that you can control enzymes. Alright, so there is competitive and non-competitive inhibition of enzyme activity. Alright, so looking at competitive inhibition. Think about what that word means, competitive. Well, that means that something is competing for the enzyme's attention. All right, in this case, you would have your normal, for example here, you would have your normal substrate, all right? And what it's doing is it's competing with your normal substrate. All right, so you have a competitive inhibitor is competing with your normal substrate for the active site. All right, so this competitive inhibitor, it looks very similar to the substrate, but it's not. Because when that competitive inhibitor binds, it blocks the normal substrate from binding to the active site. And as a result, that enzyme isn't contributing to increasing the rate of a chemical reaction. Right? It's basically that enzyme is now inert. It's no longer functional. It's taken out of the picture. All right. Now, non-competitive inhibition involves what's referred to as allosteric inhibition. All right. So you have some other molecule, but in this case, it's not targeting the active site. It's targeting what's referred to as the allosteric site. Alright, so an allosteric site is just some other site on the enzyme. Alright, and in this case, the allosteric site is where this particular molecule, the inhibitor, is binding. And when it binds, the active site of the enzyme actually will change its configuration, its conformation, its shape. And the substrate can't bind to the active site anymore. Alright, so basically you have a direct way of inhibiting, and in here, non-competitive is more indirect method. Alright, so looking at regulation of enzyme activity. Alright, so we were talking about activation and repression of enzymes, and enzymes themselves, when they're produced, they can be produced in a repressed state. Alright, so in this case, if you have an enzyme, and it's inactive. It's produced by the cell, but it's inactive. It can't bind to its substrate. Alright, the active site isn't at the right shape or configuration to bind to its substrate. Alright, so these guys won't fit. You have some other molecule that's an activator that binds to the enzyme that allows for the active site to assume its proper configuration. So now it can bind to the substrate and the enzyme is activated. Here, if you have an enzyme that is functional, alright, and you want to turn it off, alright, you can have a repressor molecule come in and bind to another site of the enzyme. It causes the shape of that active site to change. Alright, so this would be allosteric inhibition, and now it can no longer bind to the active site. Your substrate can no longer bind to the active site, so that enzyme is no longer functional. So enzyme is inactive. Alright, so let's look at an example of competitive inhibition. Alright, so as it applies to bacteria. Alright, so your bacteria require the ability to synthesize folic acid. Alright, they have to synthesize it. Alright, so this is the biochemical pathway for folic acid synthesis. Alright, so tetrahydrofolate is what we call folic acid. Alright, so when you're talking about a biochemical pathway, recall that each step in that pathway, there's an enzyme that's involved in each step that regulates each chemical reaction, is involved in each chemical reaction. So, tetrahydrofolate is basically a precursor for these guys here. Alright, so thymine, guanine, adenine, nucleotides. Alright, so the ability of bacteria to synthesize, DNA, to synthesize ATP, RNA, ability to repair DNA that's damaged. Alright, they need to be able to make these nucleotides. Alright, so normally, notice here, in this first step of this biochemical pathway, we have some precursor molecule and some product of the first reaction, precursor 2. And PABA is the normal substrate for enzyme 1. Alright, so that's what's going on here. Alright, PABA binds to enzyme 1. And it allows for precursor 1 to produce precursor 2. Alright. Now, we have antibiotics, things that we call, for instance, sulfa drugs. Alright, so sulfonamides, sulfamethoxazole. Alright, so these particular drugs are structural analogs of para-aminobenzoic acid, or PABA for short. And notice that these antibiotics, they have structural similarities, right? Because to the actual target of that enzyme. Alright, so this group here, this group here, this group here looks like this one there. And that chemical group is the one that actually binds to the active side of the enzyme. This other group out here is, alright, is important, but it can change. Alright, the big key point here is what's actually binding to the active site. Alright, so if you have a sulfonamide, for instance, this guy here, and it binds to the active side of the enzyme, alright, PABA can no longer bind. Alright, and as a result, if PABA can no longer bind to its enzyme, alright, this enzyme is inactivated, alright, basically halting that process, and the bacteria can't synthesize folic acid, it can't synthesize these nucleotides, so guess what? It kills bacterial cells. Alright, so this is an example of competitive inhibition. Alright, so there are other ways of inhibiting enzymes. Alright, so here you have you have what we call negative feedback inhibition. Alright, so what's going on here is you have a metabolic pathway. You know, your reactant molecule A is going to result in product B. Enzyme 1 is involved in that particular reaction. Alright, now product B becomes the reactant of the second reaction, forming product C. And product C becomes the reactant of the third reaction, forming product D. Now think of this as an assembly line. Alright, and basically the ultimate goal is to produce this final product. Alright, now once once a cell has met its, let's say, quote unquote quota, no pun intended, that once it's made what it needs to make, right, at a certain level, it's pretty inefficient to make more of it than what you need, right? Or let's say that you're a little bacterial cell, and let's say you make something like, let's say you synthesize a sugar, or you synthesize an amino acid, all right, something along those lines. And let's say that you come across it in your environment. Well, what's easier for you? To make it yourself, or to take up what's freely available in the environment? Well, it makes more sense to pick up from the environment what's already there. Alright, so in this case, this product D, which is the ultimate goal of the process, can actually function as an inhibitor. And it can inhibit one of the initial reactions. Alright, so coming back to the first reaction, your product can come back to enzyme 1. Alright, so basically product D can come back and target enzyme 1. And in this case, it acts as an inhibitor. It turns off this enzyme because it inactivates it, and now you stop this chemical reaction from taking place, meaning that you stop this reaction and this reaction. Now it can be turned back on, alright, because product D can decrease the concentration, and then basically that inhibitor will plop off the enzyme and the reaction can begin again. Alright, so here, giving you an example. Alright, so you have aspartate. Alright, is your initial substrate molecule. Alright, it binds to its respective enzyme, and you form this intermediate. Alright, so aspartate binds to enzyme 1 here. You form this intermediate A. Alright? That's the product of the first reaction. Intermediate A can come in and bind to enzyme 2. You get the production of intermediate B. Intermediate B can come in and bind to enzyme 3. You get the production of intermediate C. It comes in and binds to its respective enzyme. You get the production of this intermediate. And then finally, you get the production of the end product, threonine. Right, another amino acid. Now, threonine, this is the ultimate product. Now, basically, as you build up an excess amount of this particular end product, it could come back and target the initial enzyme, bind to the allosteric site, and inactivate the enzyme, so that now aspartate can no longer bind to its active site. And as a result, the entire pathway, at least from this initial reaction, is shut down. Alright, this is kind of giving you some other examples of feedback inhibition. Alright, so here your end product is targeting your first reaction. Alright, so now enzyme A can't allow for the production of intermediate 1. Alright, so that's a very simple linear pathway. Alright, now here, alright, this feedback loop is a little bit more complicated, alright, because product D can be used to produce two products, either AA1 or AA2. Now, AA1 can inhibit this reaction here, D being converted to X, by targeting enzyme X. AA2 can inhibit the reaction of D being converted to E by targeting enzyme D. Now, what if, for instance, the cell produced an excess amount of AA1? Alright, and let's say that it targets enzyme X and inactivates it. Well, what does that mean? Well, that means that this pathway is shut down and this process is shut down. But this pathway is still going on. So this reaction, product D is diverted into making this final product, while this side up top is turned off. Alright, so this is kind of illustrating again this idea of whether you have low production of a product versus a high production of a product. Alright, so if the cell is producing a product at or below its quota, alright, then it's going to continue to produce this said product. Alright, so it's producing enough that it remains under the radar. But when a cell starts producing too much of it, alright, that's when this product can come in and bind to the allosteric site of the enzyme. It causes the active site of the enzyme to change its shape. It no longer is able to bind to its substrate, and the pathway for making the product is turned off. Now, you can classify enzymes based on their name. Alright, so in this case, the name of the enzyme tells you the type of chemical reaction that it's involved in. Alright, so here are a couple examples of enzymes. Alright, so this is just kind of introducing you to this idea that the name of the enzyme can tell you about its target. It can tell you about the type of biochemical reactions it's involved in. Okay, so an oxidoreductase. Well, we've already talked about oxidation reduction. Well, an oxidoreductase enzyme is involved in the transfer of electrons from one substrate molecule to another. A dehydrogenase enzyme, dehydrogen, has something to do with the removal of a hydrogen from one molecule and it being transferred to another. A transferase is involved in transferring a functional group from one molecule to another. Alright, a hydrolysis or hydrolases are involved in breaking chemical bonds using water, water, lysis or lysis, breaking. Alright, your lyases, you add groups or remove groups from double bonded substrates. Your isomerases, alright, so isomerases, key word, isomer. Alright, so what is an isomer? An isomer is two molecules that have the same chemical formula, but they look completely different. Alright, so you alter a molecule from one isomeric form to another. And then you have ligases. Alright, so ligases catalyze the formation of bonds with the use of ATP and the removal of water. Alright, so ligases will be important when we're talking about DNA replication, transcription, translation. Alright, so here you're looking at an example of a oxidoreductase enzyme, so on the left. Alright, so here you have, for example, cytochrome C oxidase. Alright, so this particular cytochrome C oxidase, this is a protein, membrane protein, that you find in the membrane of some bacterial cells. It'd be something you'd also find in the membrane of the mitochondria. Alright, so the whole function of this cytochrome C oxidase is it's involved in oxidation reduction. Alright, so transferring of electrons from one molecule to another. Now in this case, you have cytochrome C oxidase transferring electrons from the protein here to oxygen. Alright, so this is the final step in the electron transport chain, where these electrons are transferred to oxygen and you get water as a byproduct. Alright, so if you recall looking at the chemical formula of cell respiration, that one of the final byproducts is water. So this is where the water is coming from, is this chemical reaction that occurs in the membrane of the bacterial cell, or if you're talking about the mitochondria, in the inner membrane of your mitochondria. Now, here on the right, you're also looking at another type of enzyme, a dehydrogenase enzyme. Alright, so a dehydrogenase enzyme, as the name implies, dehydrogenase. So you're removing a hydrogen from a molecule. So here, lactate dehydrogenase. Alright, so this is telling you two things. One, it's telling you the substrate. Alright, in this case, the substrate of your enzyme is the sugar lactate. And the activity of that enzyme, what it's going to do. So it's dehydrogenase. So it's going to remove a hydrogen from, or hydrogens from lactate, which is what takes place. You get the hydrogens that are removed from it. Alright, so that hydrogen, poof, gone. They get transferred to NAD+, and you get NADH+. Alright, so this is actually one of the steps that takes place, alright, in fermentation. Alright, where your lactate gets converted back into pyruvate. Alright, so next we have an aminotransferase. Alright, so your transferase enzymes. Any type of transferase enzyme is going to tell you that it's going to transfer some type of functional group. Alright, well here it's telling you what's going to take place. You have an aminotransferase. Alright, so you're going to transfer an amino group from one molecule, one biological molecule, cellular molecule, and transfer it to another. Alright, so aminotransferase, you have alanine with a amino group, and you're going to transfer it to this other molecule. Alright, so alpha ketoglutarate. Alright, so the amino group gets transferred to alpha ketoglutarate, you're left with pyruvate. Notice pyruvate. The difference between pyruvate and alanine is that the pyruvate's missing the amino group. And then here, your alpha ketoglutarate now is glutamate, and it has your amino group. Alright, so this enzyme was involved in the step of transferring the amino group from one molecule to another. Alright, so next, looking at your hydrolase enzymes. Alright, so hydrolases, as the name implies, hydro meaning water, lase or lysis. Alright, so if you recall, when you're talking about taking a larger biological molecule and breaking it down, alright, so this involves hydrolysis reactions. Alright, so hydrolysis reactions involve enzymes called hydrolases. Alright, so here you have polyalpha D-glucose as an example, and you add water and a hydrolase enzyme, and you're left with residues of maltose that get clipped off. Alright, so you take this really long polysaccharide of glucose and you start clipping off disaccharides of maltose. And all it involves is breaking of that disaccharide off through a hydrolysis reaction. Alright, so another thing would be like a triglyceride. Alright, so remember that a triglyceride involves a glycerol and 3 fatty acid tails. Alright, now those fatty acid tails are connected to the glycerol molecule by a covalent bond called an ester linkage. And you have an enzyme that comes in, a hydrolase enzyme that comes in, lipase. Alright, it's a type of hydrolase enzyme that comes in and breaks apart that ester linkage so that it detaches your fatty acids from the glycerol backbone. Alright, so this is actually a process that's going to be involved in something called beta oxidation, where you actually start to utilize fatty acids as a fuel source. Okay? So, next, let's look at lyases. Alright, so your lyase enzyme. So your lyases are involved in basically, for instance, you have like pyruvate decarboxylase. So pyruvate decarboxylase, you have pyruvate, and you're basically cleaving off chemical groups here. So you have pyruvate decarboxylase. Well, that tells you that you're going to decarboxylate or remove a carboxyl group. Alright, so pyruvate, you have a carboxyl group here. So basically all you're doing is cleaving that off the molecule. Alright, you're cleaving that functional group off. You're left with acetaldehyde, and this carboxyl group is nothing more than CO2, carbon dioxide. Alright, so here you have another lyase enzyme. So you have isocitrate, all right, which is actually isocitrate. It's an isomer of citrate, all right. Now, here isocitrate lyase takes an isocitrate and breaks it apart into here succinate and glyoxylate. Alright, so basically this part of the molecule here becomes your glyoxylate. This molecule, part of the molecule here, becomes your succinate. So basically it just cleaves a part of the molecule. Alright, your isomerases. Alright, so your isomerases are going to take one form of a molecule and convert it into its isomeric form, another isomeric form of the molecule. Alright, so here you take your alpha D glucopyranose form of glucose 6-phosphate. Alright, so basically it's a fancy way of saying that this molecule is in a ring structure. Alright, so the pyranose is telling you it's a ring structure. Alright, and then you convert it into its isomer form, or not its isomer form, but it's linear open chain form. Alright, so it's one big linear molecule instead of being in a chain. And you have glucose 6-phosphate. You have the enzyme glucose 6-phosphate isomerase. So an isomer of glucose 6-phosphate would be fructose 6-phosphate. Alright, and all that's taking place is that this little bitty carboxyl group here goes from being at the end of the chain. Alright, so you're talking about, for instance, an aldose sugar, and you're going to convert it into here, switch it so it's in the backbone of the molecule. Alright, so this is a ketose sugar. Alright, so basically these are what we refer to as carbonyl groups. Alright, so glucose has a carbonyl group at the end of the chain. Fructose has it in the middle of the backbone. Alright, so here all you've done is you just swapped it from a aldose to a ketose sugar. So you end up with fructose. And then you have the new ring form of fructose 6-phosphate. Alright, so you go from one isomer to another. Alright, so talking about ligase. Alright, so ligase is important in DNA replication and transcription and protein synthesis. Alright, so here you have DNA ligase. Alright, so you have your growing DNA strand. Alright, so this is your pre-existing DNA strand here. And remember that DNA is always going to be assembled 5' to 3'. Okay? So here you have a 3' hydroxyl group that's going to be attached to your growing backbone of the molecule. Alright, so here you have your pre-existing DNA strand. You have your growing phosphate attached to its DNA strand. Alright, and basically this hydroxyl group that's free hanging attaches to this phosphate. Alright, so this end is 5'. Alright, and then this end will be 3'. Alright, so it's being sized in this direction. Alright, so you have a ligase reaction between this hydroxyl and this phosphate. Alright, so that's what you have here. Is you have your DNA ligase, you have ATP hydrolysis that supplies the energy to create that covalent bond, that phosphodiester bond here. Alright, so that's what you're seeing here. The reaction you're seeing, alright, here you see, alright, over here. Alright, where you have your growing DNA chain, so you have the 5' end here, and it's being synthesized in the 3' direction. And so here you have your 3' hydroxyl on your pre-existing DNA chain. You have your phosphate on your little bitty nucleotide that's going to be attached to it. And so this phosphate gets attached to that OH, to that oxygen, and you get your new covalent bond, your phosphodiester bond, that now holds together this pre-existing nucleotide chain and the addition of this new nucleotide. Alright, so you can also classify enzymes by their functions and the types of molecules that they target. Alright, so a carbohydrase targets carbohydrates. It is involved in, since you have the name hydrase, right, it's involved in the breakdown of these particular biological molecules. Alright, you have amylase that targets starch. Alright, so here you have starch that's broken down by amylase into its disaccharide units of maltose, for instance. You have maltase, which targets maltose, that breaks down the maltose into into 2 glucose monomers. Alright, so you have an enzyme specific to sucrose. Sucrase breaks it down to glucose and fructose. You have lactose. You have lactase as the enzyme targets lactose. You have glucose and galactose as products. You also have proteinases, proteases, peptidases. Alright, all of these target proteins. Alright, so you talk about digestion of proteins, you have proteases and peptidases that target the peptide bond that hold together your amino acids. And they cleave very specifically certain peptide bonds between certain amino acid residues. Alright, so that's what's going on here. You have protein digestion that involves these guys. Then you also have lipases, alright, which we talked about previously, that target, your fats, your triglycerides. You have DNases that will basically do the opposite of this reaction. Instead of lengthening the DNA one nucleotide at a time, what happens is you start actually doing the opposite. Your DNases will come in and actually cleave these phosphodiester bonds and break apart the nucleotides. And then you have synthases or polymerases that actually polymerize and make these smaller monomeric units of like sugars, for instance, or amino acids or nucleotides, and string them together into longer strands, alright, making your polymer. Alright, so you have something like DNA polymerase. Well, DNA polymerase polymerizes nucleotides into your nucleic acid of DNA, your polynucleotide. Alright, so which of the following mechanisms of enzyme control blocks a reaction catalyzed by an enzyme by binding of a product to a regulatory site on the enzyme? Well, you have enzyme repression, you have competitive inhibition, you have enzyme induction, you have non-competitive inhibition. Alright, well here what you have is some other product that's going to somehow compete, not necessarily compete, but it's going to regulate the enzyme and how it functions. Alright, so it's not competitive inhibition. Reason being is you're not competing for the same active site. Alright, You're not inducing because you're actually turning off the function of the enzyme, right? So binding to a product to a regulatory site on the enzyme. Well, in this case, what you're actually doing is you're repressing the action of the enzyme.